Okay. Okay. Are you guys able to see my screen?
Yes, sir.
Yes. Okay. Okay. So I will just quickly go over the multi-taper method one more time. Fourier analysis convolution. Where is multi-taper? There. Okay. So the idea behind the multi-taper method was that this method allows you to have multiple windows or what are called tapers that can be multiplied with the signal, and these tapers are all orthogonal to each other. So they look like this. If you've got multi-taper, so you say I want 4 tapers. So these would be your 4 tapers. If you take a dot product of any of the two here, you'll get a 0. So then when a signal is multiplied by these tapers, and then you compute its spectrum like this. So from the same signal, you can have now multiple spectra, and they are independent of each other because the windows are orthogonal. So you can now average these and get a much more sort of smoother or spectrum which has lower variance. So this comes about because the spectral estimators are known to be very noisy. This is something I had discussed without really telling you why that is the case, because it's quite involved. But we saw that the standard deviation divided by the mean of the power was close to 1, and it followed an exponential distribution. Then if you do the multi-taper analysis, your total variance goes down by a factor of n, where n is the number of tapers that you have. Okay. For the amplitude, it's going to be root of n. So you get a more and more smooth estimate, which has a much higher reliability or much lower variability. And you can choose how much smoothing you want to do. So you are sacrificing your frequency resolution in order to make your estimate much more reliable. So this is what happens when you do the multi-taper analysis on the LFP signal that we had during the stimulus period. For a small number of tapers, you see this nice bump, and then it sort of fades away. You can barely see it as your tapers increase. And if you had lots of trials like what we have in this case, multi-taper is useless. You really should use just 1 taper or 2 tapers if you've got like 100 trials. But if you only had 5 trials or 10 trials, where the spectrum otherwise would have been very, very noisy, and in that situation, sacrificing a little bit of your frequency resolution by increasing the number of tapers. This number here is the time frequency of TW, whose minimum value is 1. But multi-taper allows you to choose numbers that are greater than 1. Essentially, this is the reduction in frequency which gives you a lower standard deviation or a better estimate of the uh, spectrum. Okay. So, are there any questions on this multi-taper method? So I was going to show you a little bit on MATLAB. Remember, you have to get this package called Cronux. Oops, not here. Um, multi-taper. Yeah, you have to download this package called Cronux, which it's a very popular software that is used in neuroscience. And once you have it, you will find this command called empty spectrum C, which gives you the multi-taper spectrum. And you can see that the command itself is very, just a few lines. That's it. Um, so what they are doing here is generating the tapers. This command generates the tapers, and then it computes the MTFFT, Fourier transform of the tapered signal. So I just wanted to show you, this is basically how you learn about multi-taper in MATLAB. So if you open mtspectrumc.m, so if you do edit spectrum C, so then it will open this, and there you can check all the commands. And a simple strategy is to just put a breakpoint where you want. So if you click on any of these, it creates a breakpoint. And then when you run the command, it will just come and sit here. And then you can see what each command is doing because will be able to see all the variables here. So I just wanted to show you a little bit about multispectrum, the tapers. So let's say I say I have n of 1,000 and sampling rate is 1,000. So I have specified params.fs and params.tapers. Empty spectrum requires this variable called params. So let me just actually go back here and see if I have put it somewhere. Yeah, here it is. Here you can specify the tapers that you want, and it takes this variable here TW, which could be at least one or could be a larger number, and K, which is the number of tapers that you want, the maximum of which is 2 TW minus 1. So this could be 1, 1, 2, 3, 3, 5, and so on. There are some other options here, not very, so whether you want to zero pad a signal or not, I'll tell you what that means in a bit. Um, and some other things. So I'm just setting params.tapers and params.fs here and taking default values for the rest. So let me just run this much. Control C, Control V. Yeah. So params now is going to take 1,000 here. Let me just put params.pad at 1. Minus 1 means no padding. And let it take default values for the rest. Params. Yeah, okay. So then this DPSS CHK, which is the command here, this is the one that generates the tapers. Okay, so I will run this command and I can now plot taper wins. So this is the multi-taper window when you only have a single taper. Okay, so this is nothing but windowed Fourier transform now, where the window here is the Slapian window. And you can see that it doesn't look much different from, let's say, a Gaussian window or Hanning or Hamming window and so on. They look quite similar. I could plot the absolute and then it looks like this. You can barely see it here. So I'll do this on a log scale, log 10. This is the window when you had a single taper. Okay, and you can see, so it's symmetric about 500 because the sampling rate is 1000. So let me just do slim 0 to 100. So the window here, the main lobe, and then there is some sort of side lobe. It's hard to see it in this kind of a plot. So I'm going to increase now the number of tapers, and then you can see it a little bit better. Okay, so this is my 0 to 500 hertz. This is frequency for a window which look like this. Okay, so now I'm going to increase the number of tapers. So let me say I'll have 3 tapers and give me, 3 TW is equal to 3 and give me 5 tapers. So now let me run this command again. And now the size of taper wins is going to be 1000 into 5 because we have 5 tapers. So if I run and plot the first taper again like this. So now you can see that if I choose this TW equal to 3, the taper is much narrower in time, right? And because it is narrower in time, it's going to be wider in frequency. Okay, so I guess, well, I removed the previous one. So let me just do this. I'll say 1, 1 here and call this taper wins 1. And now I'm going to do, let me even do larger, 5, 9 and call this tapers 9. Okay, tapers 5. So clear figure. By the way, is this font size okay for you guys? Are you able to see what I'm typing or is it too hard?
No, I can see.
You can see, huh? Very good. Okay, so subplot 211 and plot taper wins 1. This is when you had a single taper and now you say, hold on, plot taper wins 5, plot the first one in red. This is the first taper in the multi-taper method when you chose TW equal to 5 and you have 9 tapers at your disposal. So I could also plot the second one. Let's say green. See, this is going to be now orthogonal to the red one. And then 3 would be, say, black. This again is orthogonal. So these are now the multi-taper windows. Okay, so each signal, whenever you have one trial, you are going to multiply it with each one of these. And that will give you one windowed signal. And here in this case, you will take the average of those 9 windows. So now if I look at the spectra here, so subplot 212 and then plot abs 50 taper wins 1. Sorry, so I'll do a log 10 here. So this is when you had a single taper. This is the Fourier transform. So X slim 0 to 500. And what happens now if I look at the first taper of taper wins 5, which is again the multi-taper with TW set to 5. So hold on here and plot log of the absolute value of the FFT of the and plot it in red color. So notice what has changed here. Um, well, anyway, so let me plot all of them and then you can appreciate this better. The second one is now green. The third one is black. And although it's harder to see here, if I extend from 0 to 50, let's say, you can see that the red, green, black, these have a much larger width than this one. This one tends to fall back, this blue one, pretty fast. These guys have a larger width. So this is this time frequency bandwidth. And between these, the sideband is smallest for the red one. And then this sort of starts to climb up. The side bands start to become bigger. This is the same effect that we had seen here. This being so narrow, and then the side bands starting to become larger and larger. Now let me do a really large number of tapers here, and it will become much clearer. So suppose I say my time this is 20. Okay, so 20 hertz is now TW is 20, and let me take 20 into 240 30 tapers, and I'll call the 20, and I'll run this. I'll go up here and say plot. What was it? Taper wins 20, the first one, and let me plot it in cyan. Okay, so see, it has become even more um, narrower in time, and if I plot its log 10, 20. Look at this now. This is now starting to resemble what we have seen in the figure. Can you guys see the cyan, or maybe I'll just plot? It has now huge lobe here. Now, this is now starting to remember in the here now. Um, and you can see that the first one now has a much bigger lobe. So it's almost about 20 hertz. So you can see, because TW is set to 20 and my frequency resolution here is, yeah, so it's 1 second here. So it's 20 times worse than the best resolution, which is 1 hertz. So the lobe is about 20 now. So if I do a multi-taper estimate with this, my frequency resolution is going to be very poor. This doesn't mean that I will have frequency, samples separated by 20 hertz. I will have samples separated by 1 hertz, like before, but they'll be really smooth versions of each other. I mean, so the spectrum is going to be extremely smooth, which means that by looking at the power at one value, I can predict what the neighboring value is going to be. So in that sense, it has poorer frequency resolution. Okay. Just for, yeah, I'll do another one. So tapers 20. Let me look at the 10th one. And again, I will do, I'll just do K again. Oops, what happened here? Uh, Huh? Where is the black one? 1 second, I'll just clear the figure. Subplot 212 plot taper wins. Oh, I was plotting the actual signal. Sorry, I should have plotted the log of the. Yeah, this is what I had shown earlier. Now this is for 500, the entire duration. So now I'm just going to look at 100 hertz. And now if I hold on and plot to the 10th one in black. So this is how it's going to look like. And it gets worse and worse the higher up you go, okay? So as your taper number increases, you can see that the side lobes are getting bigger and bigger. So eventually this taper will not have much of spectral concentration, only limited to 20 hertz. It's going to be everywhere. So then it's not a useful taper for us. Okay, so that's why when you do multi-taper analysis, you only choose the first few, and the maximum limit that is given to you is 2 TW minus 1. Okay, so that's that. I wanted to show you guys how the multi-taper windows look like, and also, encourage you to actually just run these commands and play with the signal. You can then multiply, generate some random signal also, and see how windowing affects the frequent the spectrum and so on. So that will give a very good idea about how all of this works. Okay, any questions on this? So I'm done with multi-taper. I'm now going to move on and start time frequency analysis. If there are any questions, please let me know. Okay, so let's move on now. Um, so by the way, I just wanted you guys to update a little bit about our plans for the rest of the course. So today is 19th April, and this is the 22nd class now. You have an assignment due this Sunday, 25th. And after that, you will get another assignment, which will be the MATLAB part of the LFP. So I think we will need about 5 classes more to finish the LFP segment. So that will take us to about 26 classes or so. And for EEG, I need at least 5, 6 classes. So we'll actually be finishing up just between these LFP and EEG. So the classes will end in about by about mid-May, about 15th of May. So remember, this is a 2 credit class and 1 credit of this grant proposal. So we are supposed to have only 32 classes, which is what we have had in the past. This time I'm going a little bit slowly, so we'll be not be able to cover some of the things that I had thought of, but it's okay. I think we are, the pace is fine the way it is going right now. So after 25th, you will immediately get another assignment, and we will also start talking about how you are to write your grant proposal. That is going to take a while. I mean, at least about a month or so. The harder you work, the better it's going to be. So after your third assignment, you will get an assignment on EEG, and those will be the 4 assignments for this course. And you have to also start thinking about your grant proposal. which will then be due when the semester ends. So that will be like early June or end of. When is the last? When is the semester officially ending? What is the last day for exams and that? Does anyone know? Um, so our semester started in February, 22nd February, I believe, was the first day of classes. 22nd March, 22nd April, 22nd May. So the classes ought to go on till at least end of May, I would think, 3.5 months. And then the exam time starts. So during that time, we will just have the grant proposal, and that would be it. Okay, so that's the approximate time frame. Okay, so now let's go to time frequency analysis. Here I will not do a very detailed mathematical sort of description, but it will more be along the lines of the second part of the course that we have done, where we have just shown signals and try to understand how, so like the intuitive feeling of how the different algorithms are working. So there's a paper that we wrote a few years ago in Journal of Neuroscience. It's a review paper of all the different time frequency analysis methods that are used in neuroscience. So if you read that paper, along with that paper, there's data and codes that are provided. That's on GitHub in this. So all you have to do is just work with this, and it will then, I'm hoping, will make a lot of sense. So we are now tackling the second part of this three-part problem that we had posed earlier. We had first asked, are there any oscillations in the LFP? And we got our answer. The answer is right here. This bump here is the gamma rhythm. So yes, there are gamma oscillations, and there's a reduction in low frequency power also when a stimulus is presented. Now the question is the power or frequency of these oscillations, if they are present, are they changing with time? So we now need to understand how the power varies over time. And that's the question that we are going to now address. Okay, so the simplest thing that you can do is what is called short time Fourier transform. And the idea here is very simple. You've got the signal here, xt, which is this long signal like that. You usually multiply it with a window and then take the Fourier transform. That becomes your windowed Fourier transform. And here's a situation where I'm taking the window ht, but then I'm translating that window by some amount shown here. So it gives me lots of different segments, and I'm computing the FFT of each. So it's essentially taking the Fourier transform of a short time segment, hence the name short time Fourier transform. So this is the formula for this. It's now going to give you an output x, which is not just a function of omega, but it's a function of omega as well as tau, where tau determines where this window is placed. Okay, so conceptually it's very straightforward. It's just windowed Fourier transform, but the window is placed at different locations, and you generate a Fourier transform for each location, and that's your short time Fourier transform. So let's see how the short time Fourier transform looks like. So this is my single trial, one LFP signal. Time 0 is when the stimulus comes on, then there is depolarization and so on. The stimulus is only on for 400 milliseconds in this case. This thing is the short time Fourier transform. What is different between this and that is the length of the window that I'm using for computing the Fourier transform. Okay, so here I have a long window, 256 milliseconds. So this window is stretched out in time. It's a longish window. And then I can make it shorter and shorter and so on. So at each location, I'm placing the window and I'm computing the Fourier transform. So I'm getting a vector for each time value. And then I look at the next window, which is shifted by some value and then computing a vector. So these are two-dimensional plots where the time axis is shown on the x-axis and frequency is shown on the y-axis. And now the power of the signal or amplitude of the signal is showed by a colored plot, by a color. The more reddish the color, the higher is the power. Okay, so yeah, this one doesn't have a color bar, but anyway, so reddish colors mean more power, bluish means less power. So the short time Fourier transform of this trial is going to be going to look like this. And you can see it's hard to make sense of anything because a single trial estimate is very noisy, as we know. If you reduce the window size, you will get better time resolution. The windows will be closer to each other in time. But see now that they're becoming poorer in frequency. And that's because you're getting frequencies at farther values. These are 1 by t. So if t reduces, then 1 by t increases. And this is further, even shorter here. So much longer frequency wins. So they all look very bad for a single trial. But remember, you have got 76 trials, so you can compute 76 such, such tiled spectra, and look at the average of all of them. So here you had averaged the signal to get the ERP, the evoked response. The average of 76 such spectra like these, the average will look like this. So by the way, is this spectrum here the Fourier transform of this signal? Yes or no? Okay, so there are two answers, yes or no. Please say one. It's okay if you are wrong, but odds are you'll be right with 50% probability.
Could you repeat the question?
The question is whether this spectrum that you see here, the short time Fourier transform of this signal here. This is the short time Fourier transform of this signal, and there are 76 such trials. So you'll get 76 such spectra like these, and then you are averaging. This is the mean of these 76. This is the mean of 76 such trials. So I'm asking whether this is the short time Fourier transform of this signal.
I don't think it is, but I mean, this is
Why not?
probably we kind of covered a bit of this in time frequency analysis class, so I have a bit of a hack over here, I guess. But I mean, it's got something to do
Okay,
with the
so
fact.
then let the others answer, then we will proceed. Okay, others have to guess.
Not
And I guess,
exactly.
huh?
Not exactly. Is
Why
it
not?
equivalent? The average of Fourier transforms
Hmm.
has to be the inverse Fourier transform of the average of the signal. So I don't think so. It is equivalent.
No, that argument is not valid. Fourier transform is linear. If you take the Fourier transform of a bunch of signals,
So in that sense, if we have the since Fourier
average of
transform is linear, then we can we assume that it is matlab, then to it should be the Fourier transform of the signal. Well, sorry, yes, the answer should be yes then.
Srishti thinks it's yes. This is the average of this. Anyone else with a differing opinion? Okay, let me say the answer is no. So, can you think why this is not the case? Why is this not the short time Fourier transform of this? Actually, this question could have been asked even for, for these. We don't really need to talk about short time Fourier transform. It's the same for, yeah, here. So is this the Fourier transform of the ERP? So each trial has given you, right? So there. So this is one trial. That trial has the Fourier transform that looks like this. Right? So if I have 76 trials, then I'll get that ERP, that nice sharp depolarizing dip that we have seen in the past. Where have we seen that? It's too far out. Yeah, we would get this black trace. Okay, so now I compute the Fourier transform of each of these. So that's now that's now 76 of this, and then I average that. So the question is this the Fourier transform of the ERP? Yes or no? Okay, I'm not proceeding until I hear answers.
Can it be restated in a more mathematical way so that I can kind of like look at it in the terms of equations, maybe like
Yeah, you
so
have all the math. Here's the math.
so the FFT of the average of all the signals is not the same as the average of all FFTs of each individual signal.
No, it is. That's what I'm saying. We
Okay.
know that FFT is linear, right?
Right.
So, yeah, so xn, yn. So here you have x1n, y1n, x2n up to x76n. The Fourier transform is going to be the same. It's the average of here a and b is just 1 by n, right? So it's the average of the FFTs.
Okay, yeah. So it was valid in the previous case, but it's not valid in this case. That is
Hmm.
the
Why not?
They didn't.
here there is also that window which you have to look at.
No, there's no window. Just right now not reached windowing. Just the FFT. Well, can you think why you're not seeing? So here it seems that there's a lot of power at 55 hertz, right? Oscillatory power at 55 hertz. But if I look at this ERP, where is it? Yeah, you don't seem to see any 55 hertz component in this period. Why not?
Time and voltage वाले graph से तो because since we have studied that the frequencies are almost everywhere, how come we can conclude from here that we do have
No,
a
no. So that Fourier transform that with the bump that I was showing was for 200 to 400 milliseconds, this period.
okay, okay.
Well, anyone else who wants to guess? See, if it was so easy, just computing the FFT of the ERP, you really don't need to save all the data. You do 76 trials, you just save the ERP, and everything that you want to see is there in the ERP itself. So you could have just saved instead of having a 76 cross 1000 vector, you could have just saved the 1 cross 1000 vector and worked with that. Why are we bothering computing the FFT of individual segments? The answer is actually not very hard. The answer is there in this formula itself that I'm using. Okay, okay, just to make sure everybody gets the question here. The question is that FFT is linear, that we have seen over and over again here, right? So that means that if I've got 76 trials and my ERP Y is just 1 by 76 of x1 plus x2 plus dot dot. Sorry, I don't have my pen with me, so that's why the writing is so poor. 76. Okay, so this is the first segment. This is the second segment. I've got 76 trials, right? So first trial, second trial, and the ERP is just the average of that. Now, what this linearity tells you, then that if this is in time domain, if its Fourier transform is x1 omega and this is x2 omega and this is x76 omega, then your Fourier transform of y is just this. Okay. So if I give you y, you could just take the Fourier transform of y directly. And linearity tells you that it's going to be the same as the average of the 76 different Fourier transforms. Okay, so the question then is what you are seeing here. Here, which is the average of these 76 waveforms here, is it the same as computing the FFT of the ERP? The answer is no, and the answer is right in this formula. So just think and let me know why the linearity is not working here.
Well, a small guess. Here we are plotting log, and log itself is not linear function.
Okay, you got the log, but if I had done it in the linear, if I had not used log, the same thing would have held. Why? So the answer would have been, that it's not linear, even if I had taken the not taken the log.
Okay.
Okay, it's right next to the log. I'll give you more hint.
Summation over frequency. I don't understand it. But, um,
Okay, what's next to the log here?
Is it because you're plotting only the absolute value?
Yeah, exactly. That's the answer. See, FFT is linear, but FFT is a complex quantity. Here I am not showing you the FFT, I am showing you the absolute value of the FFT. The absolute value of FFT is not linear. Okay, so in particular, so suppose the stimulus onset at 0 produces a strong gamma in every trial like this. So in the FFT absolute spectrum, you will see a nice peak like that. Okay. Now when you present it the next time, it is induced, but at a slightly different location so that the phases are not aligned. Okay, so starting at a different phase where there's a dip here, there's a peak here. This is trial 1, this is trial 2. So the absolute value of the FFT will always have a peak at exactly the same frequency because you have discarded the information about the phase. You average all of these, you may get nothing because they are all out of phase and they cancel each other. But in the average of these, you will still see a nice big peak because you are only looking at the average, at the absolute value, not looking at the phase. If you had actually looked at this as a vector with amplitude and phase, then you would have seen the same thing here, that they would all add up to 0. Like here. But that's not what you are doing. You are looking at the absolute average of the uh, absolute average of the absolute value of the FFT. Because using this absolute value, as long as it's present, even if it is not phase aligned, averaging doesn't remove that component. Okay, so remember always that FFT has a phase and an amplitude, but whenever we show it, we always show the amplitude part We don't really show the phase. We'll do some analysis about the phase, but it's not very well. It is yeah. So it's a different type of analysis called coherence analysis that we'll do later on. So in fact, there's what is called an evoked component and what is called an induced component. So the evoked component is essentially which is phase locked to the stimulus onset. So it's always coming at the same location in each trial, which is why when you average it, you get the evoked response. That was this response here. This is the evoked response. So it is in fact called the evoked response for that reason. But in each of these trials, 76 trials that you see here, there is an induced component also, some gamma rhythm, but it's coming at different phases in different trials, which is why when you average it, you don't see it here. And if you have just looked at the FFT of this signal and averaged those FFTs, you wouldn't have seen it either because FFT is linear. So the FFT of the average of the FFTs of a signal is the same as the FFT of the average of the signal, which doesn't have this component. But that is not what I'm doing. I'm averaging the absolute value of the FFT, not the actual FFT. It's the absolute value of the FFT. This FFT will always show a peak, the rhythm, if it's present in each trial, irrespective of what the phase is, because I'm removing the phase. Okay, which is why you see these nice bumps in the amplitude spectrum. Like this. Okay, and which is why in this plot also, in the time frequency plot also, this is not the short-term Fourier transform of this signal. Because here also I am averaging the absolute values. I'm just averaging the colors here. Each of these colors is an amplitude and it has a vector associated with it, which is the phase. But I don't care about the phase. I'm just averaging the amplitude. And here you can see the gamma, it's coming up like this. It's faint, it's hard to see, but you can see this gamma rhythm. It's only well, I suppose here it seems to stretch out. But notice that if I use a very short window, it's very hard to see. Very hard to see. And that is because my frequency resolution is very poor. The tiles are all like, it's like paintbrush with strokes that are vertical. Here you are using horizontal strokes, so you can capture the rhythm. When you use a short window, you can barely capture it. On the other hand, the onset of a stimulus, this evoked response, has a broadband power. It's a Gaussian, right? So its Fourier transform is also Gaussian. If it was like a delta function, it would have power at all frequencies. So this thing can only be captured if you have a short window. Here it's hard to capture. So this is the time frequency trade-off, that in order to capture the rhythms properly, you need to have long windows. In order to capture transients, you need to have short windows. And no one window will do the trick for you. So you are essentially either going to have good time resolution or good frequency resolution or somewhere in between. This is a compromise between the two. Okay. So we will now look at all these. There are so a variety of different methods. Short time Fourier transform, wavelet transform, Hilbert transform, matching pursuit. They all essentially are can be thought of within the same framework here. where we think of each of these boxes as a little filter. This is what is called a filter bank interpretation. And within that framework, all of these different methods will fall in place. So that's what I'm going to do from the from next class. Please go over this paper that I mentioned. It's a 2016 Journal of Neuroscience paper, and it explains all these methods in very easy terms. And you can also look at these codes here, which will, in fact, there's a homework problem, so you'll have to use these codes to complete your homework. But please get going on this because we'll be doing this for the next few classes now. Okay, so I'll stop recording, and the class